Okay, thanks everyone for introducing yourself in the chat. I think that we've reached a steady number of participants, so I think in the interest of time, I'll get started. So, welcome to the Collaborative Science for Estuary series. I am Ariana with the Science Collaborative. Today's webinar features a multi-reserve collaborative research project in the southeastern U.S. that used a multifaceted research framework to better understand the importance of different estuarine habitat types and variable environmental conditions on shrimp populations. As always, before we get started, we have a quick introduction to the reserve system, the Science Collaborative, and this webinar series. The National Estuarine Research Reserve System is a national network of 30 unique research reserves, as shown on this map. This NOAA program works in collaboration with a local place-based partner, either a state agency, university, or nonprofit at each site. Each reserve site includes programs focused on the activities you see on the slide, including land stewardship, research, and scientific monitoring, training programs for the public and local officials, and education. Just a quick overview of the Science Collaborative. The Science Collaborative supports science for estuarine and coastal decision makers by coordinating regular funding opportunities and supporting user-driven collaborative research and transfer activities that address critical coastal management needs identified by the reserves. And this webinar series, the Collaborative Science for Estuaries series, features project teams supported by the NERRS Science Collaborative, program staff, and partners. Speakers share their unique approaches to addressing current coastal and estuarine management issues. And if that sounds interesting to you, join us for future webinars to learn about new methods to integrate experts and users into the research process and how their research and results might inform your work. So a quick note on your participation. Webinars are valuable learning opportunities for our program. Insights from speakers and participants like you can help inform how we support others in doing collaborative science, for example, in the development of future workshops or webinars, or resources such as tools, tips, and case studies to add to our guide to collaborative science. And this guide is open access. So with that in mind, we want to inform attendees at today's webinar that insights or feedback that you share, for example, in chat or via a webinar survey, may be used for program learning and development. Please note that if we do capture a direct quote from you that we would like to use for learning purposes, we will reach out to you to seek permission and provide an explanation of its use. All such quotes would be used anonymously, and we are very grateful for the opportunity to learn alongside you. A couple of housekeeping items. All attendees were muted upon entry, and we will be handling presentation-related questions via Zoom's Q&A feature. At the end of the webinar, we will invite you all to come off mute and facilitate questions and answers. In the meantime, you can enter questions for the presenters as they occur to you throughout the webinar, and we'll plan to discuss as many as we can following the presentation today. And the chat is visible to all, so please do use it to talk among yourselves and organizers to support any technical questions you might have. So now I will introduce our speakers. Joining us today are Robert Dunn, who spent 5 years as the research coordinator at the North Inlet Winyah Bay Reserve in South Carolina, during which time he also served as the PI for this project. Liam Batchelder is a PhD candidate at the University of South Carolina studying communities of estuarine fishes and crustaceans. He is an assistant professor in the Department of Biological Sciences at the University of South Carolina, where his lab studies the impacts of environmental change on zooplankton ecology. Graham Wagner is a wildlife biologist at the Shellfish Research Section at South Carolina Department of Natural Resources.
All right, hopefully everyone is seeing my slides now. Great. Thanks everybody for joining. We are really excited to talk about a small sample of the work that the Lowcountry Shrimp Collaborative has been doing over the last 3.5, going on 4 years now. So we're going to talk today about changing estuaries, shrimp, and the associated shrimp fishery in the Lowcountry region of South Carolina and Georgia. And like Ariana mentioned, you'll hear from 4 of us today, but this was a really large team, and so we'll get into some of the other project partners here shortly. So my name is Robert Dunn. I'm the PI for this project. I was the research coordinator for 5 years at the North Inlet Winyah Bay Reserve. I'm going to bookend the presentation today. I'll give us an overview and then wrap things up with a snippet of our outreach efforts. Josh Stone is a faculty member at the University of South Carolina, and he'll talk to us about some of the early life stages of animals. Liam is a PhD student who is really integral to both some of the field and long-term data analyses. And similarly, Graham Wagner from South Carolina DNR, a critical collaborator on both the field and lab components. In addition to us 4, this was a large team. We ended up with something like 25 core team members over the course of the project. And that doesn't include the 20 or more undergraduate students who contributed in various ways. But ultimately, like all NERRS Science Collaborative funded projects, this was about the reserves. So we worked in 3 NERRS in the southeast, from North Inlet Winyah Bay in the north, down to Sapelo Island in Georgia. We had fisheries management partners through both the regional council and state resource management agency, a couple of academic institutions, and we worked with fisheries extension agencies in both South Carolina and Georgia. So this really was just not going to be possible without such a large team. So we're fortunate to have a really collaborative group. So we came together in 2020 to begin to understand how shrimp populations might be responding to changing environmental conditions, really with the goal of conducting applied research on the estuarine associated life stages for these populations. We conducted interviews with commercial shrimping industry stakeholders in South Carolina and Georgia. And the long-term goal really is to build a collaborative team to be able to continue conducting applied research on this for this species that supports a multi-million-dollar fishery in this region. Quick background on shrimp life history. We have 3 species of shrimp in the southeast that are important. In South Carolina and Georgia, it's really only 2 species. So we'll be talking mainly about brown shrimp and white shrimp today. All 3 have a similar life history where adults are found in the coastal ocean, where they spawn. Eggs and larvae spend a few weeks at a time as plankton in the open ocean before they metamorphose into what we call postlarvae, which ingress into estuaries. The animals then spend an order of 6 to 8 months inside of estuaries, developing from juveniles into subadults before they migrate back offshore as adults, where they spawn again and are subject to being harvested in the commercial fishery, which operates just offshore. So we'll be talking mainly about these estuarine associated life stages. And when I think of an estuary in the southeast, this is really sort of emblematic of what that looks like. Most of the animals that that we caught and are thinking about in this project are found in somewhere along this seascape here, from the really smallest tidal creeks out into sort of open water, larger bodied estuaries. And really quickly, while our project was focused on the sort of environmental conditions that might impact shrimp populations, we've been cognizant throughout that the shrimping industry is facing a number of factors sort of beyond those environmental conditions that we're interested in. So these are things like socioeconomic factors, fuel prices, imports, et cetera, that while we're not expert in these, understanding that the industry is facing these challenges has helped us sort of contextualize our findings. So our project had 4 main components. We conducted a number of analyses of long-term data sets spanning sometimes 4 decades or more. We processed archived historic samples, again, some of which were collected back in the early 1980s. We conducted interviews with shrimping industry stakeholders through the fisheries extension programs in both states. We worked on some growth and mortality experiments at the University of South Carolina's Baruch Marine Field Lab. And we conducted a large-scale field sampling campaign across all 3 reserves over a couple of years that was led by graduate students. So we're largely going to talk about that first component, the long-term data analyses today. And to sort of get that going, I'll just point out that one of the first things that we did with this project was to start looking at swamp data. So we harvested 2 decades or more of swamp data from 10 stations across the 3 reserves here in the Southeast, and looked at trends in long-term trends and short-term variability in some key parameters, things like temperature and salinity. And really what we found is that across the region, temperature has been increasing on the order of 1.5 degrees or so Celsius over about the last 2 decades, with much of that increase happening during the winter months. And that's a really key time for both of these species of shrimp. So just keep that sort of context in mind as we move through the rest of the talk. All right, so I'm going to kick it off now with Dr. Joshua Stone from the University of South Carolina. Take it away, Josh.
All right. Can you hear me all right? Great. Yeah. And it's I was looking through the attendees, a lot of familiar names there. So hello to everyone, including some other folks who worked on this project as well. So thanks to everyone for that. All right, so just to familiarize you guys a little bit with the work that my lab has been doing, is we're mostly focused on that post-larval stage. So right there, when they're kind of coming into the estuary, they're still planktonic at that stage. So, after this, they kind of settle onto the bottom, burrow in. But this post-larval stage, they're still subject to a lot of, the environmental conditions that they have just drifting around as plankton in the water. And so they are kind of, throughout all of their life history, kind of each one of these stages in this life, in this life history that you're looking at, they're kind of doing very different things and subject to very different environmental drivers, different food webs, things like that. And so as part of this project, we really wanted to look at kind of these different life stages to try to figure out where bottlenecks in their population might be, how might climate change and temperature change be affecting post-larvae differently from juveniles, adults, or subadults. And so the area that I was focusing on was this post-larval life stage when they're floating around in the water column. Just to give you an idea of what these guys look like. So this is a preserved one. They look a lot better when they're alive and happy. But we've got, these are on the order of kind of a few millimeters to maybe, maybe like a 1/4 inch to a 1/2 inch long is kind of how big these guys are going to be. And then we'll preserve them, and then we can identify them under the microscope, count and measure them. And so, once we have those and if we want to study them, how are we going to do that? And so there's a few different ways we can do that. And so the reason, I mentioned this a little bit earlier, but, the reason that we're studying them is because they have this different ecology from what you might think of as like adults or juvenile shrimp. So they're very small, they're in the estuary, they're planktonic, drifting around. So they're in this stage for a few weeks at a time. So they kind of grow out pretty quick and settle down to the bottom. but in that, we're just trying to figure out, is that a critical life stage? Is that important in how many there's going to be next year to catch and eat? So that brings us to this question, right? How is environmental variability affecting these post-larval shrimp? So we need to catch them. And so we use this benthic sled here where we tow this net behind the boat. It kind of skims right along the surface of the bottom. Tends to be where these shrimp like to kind of aggregate. It also helps us to be able to tow a net in really shallow water systems as well in these estuaries. And so we'll tow that behind the boat. It catches all these shrimp. They end up in the back of the net, and then we'll preserve them for later study. And so, part of the strength of this project is we catch them a lot and very, frequently, and we have these huge data sets that we can pull on, right? 1 year it's warm, 1 year it's cold. It's hard to say, what's going to be impacting them until you get decades of data. And then once you have, for this project, we had about 40 years of data where we're sampling every 2 weeks across that time period. So this picture here is some of the folks who've been involved with that time series. This is in North Inlet, where we go out and sample every 2 weeks, and then we have all those samples archived. And so then for this project, we can go back, pull those samples out, ID the shrimp, and look at this kind of unique life history. And this is one of the oldest zooplankton time series of its kind in the world, and certainly the oldest one of these post-larval shrimp in the Southeast as well. So it's an incredible, unique data set that allowed us to ask and answer questions with this study that we wouldn't have been able to otherwise without this time series. So this, just to orient you a little bit, this is where North Inlet is. So it's near Georgetown, South Carolina, about an hour north of Charleston, or in between Charleston and Myrtle Beach. And so we've got our sampling site there in the red dot on the right. And so it's kind of in the middle of this North Inlet estuary. It's kind of high salinity, indicative of kind of typical estuaries in the area. And that's where we sample every time. All right, so let's get to the good stuff, the nitty-gritty data. So we went through basically those 40 years of samples, ID'd them down to species, counted them, and wanted to see what's been happening over time. There wasn't kind of a strong trend, but we did find some slight increases in abundance of these post-larval shrimp for both brown shrimp and white shrimp species. Interestingly enough, we also found lots of pink shrimp post-larvae. They're actually the most abundant of the 3 species at this life stage. But if you know anything about South Carolina shrimping, you don't catch adult pink shrimp in these systems. And so it's interesting because, you catch pink shrimp up in North Carolina, catch them south in Florida, and we're getting tons of post-larvae that are coming into this system, but they're not surviving past this life stage specifically. So that was, kind of an interesting result. And we were able to identify, okay, this transition from post-larvae to juvenile, that's the bottleneck for pink shrimp in the area. And so we've potentially got some follow-up work to try to figure out why that is. But as far as the brown and white shrimp, the post-larvae, slight increases over the 40 years in both of those at this life stage. And so, one of the other things we wanted to look at was timing. So, when are they appearing? There's, strong seasonality to some of these species where they'll spawn at certain times of the year, and then we see the post-larvae, during the, following that. And we wanted to see, is that shifting? Are they showing up earlier or later? And so, in this graph here, we have on the bottom axis, we have average winter temperature for that year. And each dot here is a different year of data. And then on the y-axis, it's basically our metric of when do the most post-larvae appear? Kind of what day of the year are there, are there the most larvae? And so you can see if you have a colder winter on the left, they're appearing later in the year. And on the right, if you have a warmer winter, they're appearing earlier. So we have this nice temperature signal to show that these shrimp are adjusting somewhat to temperature changes and their timing. And just another kind of way of looking at that is here we have basically we split the data set into two years that have warm winters and years that have cold winters. The cold winters are the blue line, the warm winters are the red, and then this is just the change in post-larvae abundance across the year with January 1st on the left and December 31st on the right. And you can see during those warm winters we get a much higher peak, lots more white shrimp post-larvae coming in. And they're clearly, responding to those warmer winters as well, and it's improving the post-larval ingress. So we were looking at sizes, or we were looking at abundances and how many there are, but we're also interested in the sizes because you can kind of use that as a proxy for maybe health or growth of these animals. And so for a lot of them, we would pull them out once they're identified, put them under the microscope and measure them. And so I'm going to be showing you this rostrum carapace length. Basically, it's a long chunk that can't bend, so it's a nice metric to measure. And we wanted to look at, how does that change across a year or over the time series? Thanks. So this is for the white shrimp. And so we can see, over a typical year, they start out smaller earlier in the summer, May and June, and then as we get later in the summer, they get bigger and bigger and bigger. And remember, these are staying as post-larvae only for a few weeks, and so we're really catching like new batches of post-larvae later in the summer. It's not like we're catching the same cohort as they've grown, because after they grow just for a few weeks, we don't catch them in this net anymore. Now they're caught in a different system. And so it's interesting that we can see this increase in size later in the summer. And so that's just another thing we want to investigate that came out of this. And then we see a different trend actually with the brown shrimp sizes. So they tend to be larger earlier in the year in months like January, February, March. And then they drop off in size and they get smaller later in the summer and then kind of get larger again as we catch them into the fall. And so again, we have a few different hypotheses about why this might be, but we certainly want to test that. And it's given us some interesting and new insights into their life history and how they might respond to changes in seasonality and temperature. So I told you about this data set. We've got lots of time, right? 40 years, great time set from this one estuary, but is this one estuary really indicative of what's happening in other places along the coast? And that's one of the things that coming into this project we really needed to address. Is what's happening in North Inlet, does it matter or correlate with what's happening in the region as a whole? And so one of the things we wanted to do was try to sample several different estuaries to compare. And then also sampling some of the different life stages as well. And so, I was looking at this one, post-larval stage, but then what happens next? What happens in other estuaries and what happens at different life stages? And so I think Liam's going to tell you a little bit about that.
Thanks, Josh. Yeah, that's exactly right. So we just heard about the post-larvae, and now Liam Batchelder, again, the PhD student at USC, and also note the Davidson Graduate Fellow at the North Inlet Winyah Bay Reserve, is going to talk to us about phenology and the juvenile life stage.
Hey, everybody. Thank you, Robert, for the introduction, and Josh for that seamless transition. Like he said, I'm going to be moving into talking about the next life stage. So Josh really focused on this planktonic post-larval stage, and I'll be talking more about these early to late juveniles, sort of shown at the top portion of this figure here. This life stage that settles into intertidal creeks and shallow subtidal habitats in each of these estuaries. So we've shown this figure a lot. I'm not going to talk much more about it. But what's important to mention again is that environmental conditions influence the habitat use across the estuarine mosaic and also influence the transitions between habitats within each of these life stages. And it's not just the conditions that they're experiencing at one given time, but also sometimes the winter before, which I think Josh talked a good bit about in his portion. So, Josh mentioned this already, but we really don't see a lot of pink shrimp at this juvenile life stage. So when I'm talking about penaeid shrimp, I'll be talking about the brown and white shrimp primarily, and I'll use this same color scheme throughout. So if you look at this top panel here, you'll see the brown shrimp in this orange-brown, and you'll see the white shown in gray. And this top panel just shows the mean catch per unit effort. So how many shrimp are occurring in each of these seines, which is the data that I'll be talking about over the year. And you can see that our brown shrimp enter the estuary a good bit earlier, April, and they really peak, quite a sharp peak in May. They stay relatively abundant into June, and then they sort of taper off throughout the year. We find them in pretty low numbers through October. White shrimp, on the other hand, occur a little bit later. They really start to appear in the seine survey in June, and they have a more gradual peak. They occur in much higher abundance than brown shrimp do, and then they stay in these intertidal habitats through November. So this shows that there is a decent bit of partitioning in when they're using these intertidal nursery habitats. And if you look at the panel on the bottom here labeled B, this is the catch per unit effort by year. So again, this 40-year time series, I'm going to talk a little bit more about it on this next slide. But each year you can see there's a good bit of variation in the overall abundance of both our white shrimp and our brown shrimp. So there are good years and there are bad years. Oftentimes these years correspond for both species. And so we've talked a good bit about the abundance. This has been studied in some estuaries across the Southeast and the Gulf of Mexico. But we were wondering about phenology. When I talk about phenology, we're talking about the temporal occurrence of natural phenomenon. And this is usually referred to as major life, used to refer to major life history events, things like births, deaths, migrations. And I'll be talking a lot about timing and synchrony. So if we look at the diagram here in the middle of the screen, timing is just the date at which some event occurs, or the central date of occurrence, whereas the synchrony is sort of the spread of dates over which an event, a major life history event, occurs. And I'll be talking about shrimp, but sort of a more tangible example of this would be something like the pollen blowout in the Southeast. So anybody who's in North Carolina or South Carolina right now, or Georgia, you've been scraping yellow pollen off your car for about a month. And really, it was a huge sort of blowout of these pine trees releasing all of their pollen about 2 weeks ago. And we've been seeing a taper off now. So sort of the whole period over which that occurs would be the synchrony, and sort of the central abundance when that pollen was thickest would be the timing. And changes in timing and synchrony, these influence resource availability. And for something like juvenile shrimp, which are both prey and predator, it also influences the predator-prey dynamics for the species. And because both white and brown shrimp are commercially and recreationally important, this has downstream effects on harvest and timing of harvest. Just like abundance, the phenology of nursery habitat use for white and brown shrimp shows quite a bit of interannual variability as well. So the objectives for this portion of the study were to determine if the phenology of estuary nursery habitat used by penaeid shrimp is changing over that 40-year time series, so from about 1984 until now, and then to link any variations in timing and synchrony that we observe to environmental conditions present both during that nursery period and in the winters preceding. All right, so to target some of these objectives, we analyzed a 38-year time series of seine survey data from the North Inlet Winyah Bay National Estuarine Research Reserve. So if you take a look at this map on the bottom corner of this, the yellow circle shows Oyster Landing Creek, where this seine survey has occurred bi-weekly in an intertidal creek pool since 1984. The red circle shows the Baruch Marine Field Lab. So this is a location that's in close proximity to the lab. And similar to the sampling location for the postlarval shrimp, this is just one location, but it's representative of a number of different creeks in this system and in systems across the southeastern United States. So shrimp are collected with a seine. All right, transfer. Shrimp are collected with a seine, they're identified, and they're measured. And so then we use those abundance calculations to calculate our phenology metrics here, timing and synchrony, which I mentioned on the previous screen, and then duration, which is just the time between sort of the central portion of their ingress. So 25% of the individuals in a given year to 75%. And we used a circular statistics approach to analyze this. It's a similar approach to what Josh used for the postlarval phenology. And the reason we use this approach is that it's particularly useful when you're looking at dates, which occur sort of more in a circle than they do in a linear timeline. So January 1st and December 31st are closer in proximity to one another than they would be if you used a linear approach. So getting at this first objective here, to look at change over time, each of these panels, I'll go through sort of one by one, but again, our brown shrimp are on the left here in this orange, and our white shrimp are in this gray color on the right side. And we wanted to just see if the timing, synchrony, and our duration have changed over the beginning of this time series in about 1984 through to 2022. And what we can see for brown shrimp is that the mean date of abundance, so this top panel, those circles, is not changing, and neither is the 25% or 75% ingress points. And that's the same pattern that we see for white shrimp. However, what we do see is white shrimp are becoming less synchronized. This nursery period is becoming less synchronized, and that's causing a corresponding increase in duration. So they're showing up slightly earlier or across a wider range of dates than they used to, and they're staying in the estuary just a little bit longer. So for the next objective, we wanted to understand what environmental conditions might be shaping some of this change that we're seeing. And because we didn't see trends across the time series for both species, what might be causing some of this interannual variability? We used a generalized additive modeling approach with stepwise variable elimination. Happy to talk more about this, but I'm going to sort of skip over a lot of the nitty gritty. What I think is important to mention are some of the variables that we tested and why we chose those. So in each of the full models, we included a suite of winter water temperature variables that have been shown in previous studies to influence the abundance of juvenile and postlarval shrimp. So these are things like the mean winter temperature, water temperature that Josh mentioned, as well as a couple different threshold temperatures. This 8 degrees Celsius number, which is used by managers in South Carolina to open and close shrimp seasons, as well as this 11 degrees Celsius threshold, and we did total days and consecutive days below 11. And this 11-degree number has been shown in previous studies to have the strongest correlation with juvenile shrimp abundance. So we wanted to test this as well for its effect on phenology. We also included a few salinity and precipitation metrics as well. So moving right into the results here, each circle in this Venn diagram that you see here on the right represents a different model. So we created independent models for timing, synchrony, and duration with the full suite of environmental variables. And the variables that were retained in all three of these models for our brown shrimp were the mean nursery salinity, so the salinity during their nursery period, as well as the consecutive days below 11 degrees Celsius during the winter preceding this nursery period. And with just these two variables, all three of our models had a relatively high explanatory variable. So around 74 or 72 to 75% deviance explained for each of these phenology metrics. For white shrimp, the story is a little bit less clear. So while the consecutive days below 11 and salinity were retained in some of the models, they weren't as clear-cut as for the brown shrimp models. So you can see our overall deviance explained is a little bit lower. So these suite of variables that we tested didn't do quite as good of a job explaining the variation in white shrimp nursery period. However, we did see this consecutive days below 11 degrees Celsius float to the top for white shrimp as well as something that influences both the timing and the duration of their nursery period. So this plot here shows the modeled response. So these are the model outputs of our generalized additive model for timing on top, our synchrony in the middle, and then duration on the bottom. And there's a lot of information in this figure, but in general, these milder winters, so if we look on the x-axis, we have low numbers of consecutive days below 11 Celsius on the left. So these milder winters lead to earlier timing of habitat use, which is more synchronized and occurs over a shorter duration for our brown shrimp. For white shrimp, these milder winters, they lead to an earlier timing of habitat use. However, we didn't see a significant influence on the synchrony or the duration. So they do come in a little bit earlier in years that have milder winters, but it doesn't seem to have a significant influence on the synchrony or duration of their nursery habitat use. So there's more to be investigated here as far as the effect of winter water temperatures on our juvenile white shrimp. Just some major takeaways that I want to leave with before I pass the baton on to Graham. The phenology of shrimp nursery habitat use is linked to environmental conditions. All 6 of our models, for both species had high deviance explained. Both species will likely continue to use estuary nurseries earlier in the year as our winters in the southeast continue to get more mild. We're seeing fewer and fewer days below this 11 degree and 8 degree Celsius threshold. We just don't have these cold winters that had high impact on spawning adults and early post-larval stages like we used to. shifts in timing for these two species may lead to increased overlap during this critical nursery period. So shrimp are becoming, or brown shrimp are becoming a little bit more synchronized, but white shrimp are becoming a little bit less. And that's an interesting trend. And any shift in this sort of partitioned nursery period may lead to increased overlap during the sort of critical stage where they're using very similar habitats and similar food resources. And as a result, management of penaeid shrimps in the southeast will need to adapt to climate-related shifts in phenology. So this is a good bit outside of my wheelhouse. I'm sure it's something that Graham will talk a little bit more. But this is one of these things that we noticed with this phenology analysis, and I think was sort of beneficial, a result of working with both South Carolina DNR and North Carolina or in Georgia researchers, as well as some of the management agencies in the Atlantic states. With that, I will pass it on to Graham. Oh, I guess I'll mention that this work has been published. So if you have more questions, feel free to read the paper or ask me at the end of the talk.
Thanks, Liam. All right, let's keep it rolling. Graham Wagner from South Carolina DNR is going to talk about white shrimp trends.
Cool. Thanks, Robert. Thanks, everyone, for coming and hanging out for a little bit and learning about shrimp today. Yeah, I'm going to share some interesting results that we were able to get from leveraging several long-term monitoring data sets and kind of reveal some subtle changes in how shrimp populations, white shrimp populations, might be affected by changing climate that might not have been evident kind of if you were looking at shorter time frames. So yeah, let's get into it. A figure you are now very familiar with. This is kind of a simplified schematic of the life cycle of white shrimp as they grow and develop and move throughout the estuary. We're going to be focusing on the juveniles that are hanging out in those small tidal creek portions of the estuary where they settle after kind of metamorphosing out of their post-larval stage. And then they move out of those shallow, small tidal creek portions of the estuary and move into more open water portions of the estuary as subadults. These are deeper bodies of water, think kind of rivers, bays, sounds. And then these subadults hang out, grow, eat, and then mature and eventually move offshore as full-blown adults to either spawn or overwinter, kind of depending on the time of the year. So given that they are using different portions of the estuary during different parts of their life cycle, we can actually target specific life stages based on where we are sampling. That's kind of critical to what we were able to do here. And so there's a handful of fisheries independent long-term monitoring surveys in South Carolina and Georgia that are targeting white shrimp in these kind of different habitat life stage combos. And a quick shout out to the kind of state agency and university programs that are running these surveys. These are an immense amount of work and lead to an incredibly rich series of data that are kind of, priceless. So shout out to everyone involved in all of these. So this map on the right shows sampling locations for 6 different surveys. The South Carolina Creek Seine and the South Carolina Creek Trawl are targeting white shrimp, juvenile white shrimp in those small tidal creek habitats. The Georgia and South Carolina estuarine trawls are targeting those subadults in those more open water portions of the estuaries. And the South Carolina and Georgia coastal trawls are targeting those full-blown adults in kind of coastal and nearshore waters. And so together, these surveys kind of all contribute to a time series that's spanning 3 to 4 decades, and it consists of about 20,000 individual collections in which we have shrimp data and concurrent environmental data. And altogether, we have on the order of about 500,000 individual measurements of white shrimp. So this is a massive data set spanning lots of time, lots of space. So then it comes down to what do we want to actually get out of this huge data set? And there's a lot of things we could look at, but we were interested in kind of looking at if there were seasonal long-term changes in environmental conditions, specifically salinity and water temperature as measured by these surveys. And I'll point out that in the interest of time and simplicity, I'm really going to only focus on water temperature data here. We didn't see a whole lot of change in salinity, but we do see some changes in temperature, so I'm going to focus on that. Second kind of objective, were there kind of seasonal changes in the sizes of white shrimp in each of these habitats through time? And then lastly, can we relate any of the changes we saw in environmental conditions to changes that we saw in the sizes of white shrimp? And we're just going to get right into it. So let's just kind of break this down. This figure shows annual rates of change of water temperature as measured by each of these surveys. And each of these panels is representing a season, summer, fall, winter, spring. Those individual surveys are on the x-axis. The left two in each panel are those tidal creek surveys sampling juveniles. The middle two are those kind of larger estuarine surveys sampling subadults, and the right two are those coastal surveys sampling full-blown adults. And the annual rates of change are on the y-axis. And let's not get hung up on kind of units here, but the takeaway is that if a point and its error bars overlap that dashed line at 0 on each of these panels, then there has not been a significant change in water temperature. If a point and its error bars are above that dashed line, there has been a significant increase in water temperature, kind of like that Georgia estuarine trawl in the summer panel in the top left. Likewise, if the point and its error bars are below the dashed line, there's been a significant decrease. And so kind of across the board, we see some general increases in water temperature. Half the surveys in the summer showed increases in water temperature, and at least 2 surveys in each of the other seasons showed significant increases in water temperature. And so I think we're all aware that it's fairly well documented at this point. Water temperatures have been increasing in recent decades. So hopefully this isn't a surprise, but it's good that we were able to pick up on that well-documented pattern. So we can look at white shrimp sizes the same way. So this is broken up the same format as the previous figure, but on the y-axis, we now have annual rates of change of the sizes of white shrimp. And the takeaway here is that in every season, we have at least one survey that shows a significant decline in kind of the average white shrimp size. And I really want to focus in on the fall because half of the surveys in the fall showed significant declines in the sizes of white shrimp. And the magnitude of those changes are greater than any other seasons. And that includes both of the surveys that are sampling for white shrimp in those open water estuarine habitats, those subadult shrimp. So something is clearly going on in the fall. And so we wanted to dive a bit deeper into what was going on there. And so we did that by fitting these generalized additive models to average white shrimp sizes at kind of an annual level. So this figure visualizes those models that we fit. And what's cool about these generalized additive models is that they can fit to curvy patterns, they can fit to linear patterns. And you'll see how that's important in a moment. On the x-axis, we have year. On the y-axis, we have average shrimp size, and each one of those dots is a point, and the colors indicate the different surveys. And one thing that I think is really cool about this figure is you can clearly see the separation of the different life stages that each of these surveys is intended to sample for, and those are kind of denoted on the right-hand side for reference. Our yellow survey there shows a significant decline in the sizes of juveniles in tidal creeks. And I do want to point out that that's likely driven by some pretty large shrimp early on in the survey. We think that might be related to some cold snaps that reduced densities in tidal creeks and then allowed white shrimp to grow a little bit bigger than they might normally. But overall, that pattern kind of declines and levels off. But I really want to hone in on the kind of pink points in line and the navy blue line. So those are our 2 surveys that are sampling subadults in kind of deeper waters of the estuaries. What's interesting about those declines is that the decline presents as a linear pattern. And so that's suggesting kind of a slow and steady decline over the entire 40-ish years that those surveys have been running. If that decline were related to a disease outbreak or a crazy weather event or some kind of event or acute cause, we would expect there to be a break point or some curve in the pattern there. But instead, these are linear declines. So that is suggesting that something slow and steady, like changing climate or increasing water temperatures, is driving what's going on here. So then this kind of begs the question, well, are the shrimp themselves getting smaller, or is it something else kind of going on behind the scenes? And so we were able to dive into that a little bit further. And we did that by essentially taking all of the individual shrimp measurements and graphing length, frequency, density distributions. And so that's what you see here. On the x-axis, we have the sizes of white shrimp. On the y-axis are these convenient little time periods. And old data are shown in darker colors towards the back of each panel, and lighter colors indicate newer data in the front of each panel. And what's interesting is we can see those declines presenting themselves in those two middle panels, those two estuarine, deep water estuarine subadult shrimp surveys. And so we can see that linear decline we talked about, but we can also see that the cause of that decline is that there are fewer large shrimp kind of populating the right-hand side of each one of those curves or each one of those peaks. And so that's suggesting that there are just less big shrimp in those open water habitats as time goes on. And that is kind of complemented by the fact that if we look on the right-hand panels, these are the coastal surveys, we don't see a change in white shrimp size. And those are the shrimp, the big mature shrimp hanging out in those coastal and nearshore habitats. So this is suggesting that the maximum size of white shrimp aren't changing, only the size of shrimp kind of in those estuarine habitats, in those deeper estuarine habitats where we're catching subadults. So this could be explained by larger adult shrimp leaving the estuary earlier in the year. So we're not capturing them, and therefore we're driving the size down. That could be related to faster growth in the estuary due to warming temperatures. It could be related to some body size cue for migration being reached earlier in the year. It's likely some combination of faster growth and shifting temperatures causing some migratory cues to be shifted. But clearly, there has been some changes in kind of the demography of white shrimp in those estuarine habitats as time goes on. And so the kind of takeaway here is that we were able to detect warming temperatures in multiple surveys across multiple seasons. And we also concurrently documented declining white shrimp sizes. Those shifts were most apparent in the fall. That's a time when historically white shrimp landings are extremely important. So there's some management considerations there as well. And these presented as kind of gradual linear declines. And so that might be explained by altered growth rates or migratory patterns, but is likely not associated with disease outbreaks or weather events or things like that. But all in all, we kind of need to put in some more work to kind of pinpoint the exact mechanisms that are driving this and that are interacting to kind of influence migratory cues. We know salinity and temperature can affect migratory cues, but so can body size. So there's some really complex interactions going there. But overall, this kind of serves as a case study for long-term impacts that gradual warming might be having that aren't evident on smaller timescales. But it also speaks to the benefit of having these long-term data sets to play with and really dig into. Yeah, thanks.
Thanks, Graham. So Graham, Josh, and Liam will be around afterwards to answer any questions. And we've got a few other things to cover here. Just to really quickly wrap up, I'm going to give some insights on our industry outreach to industry stakeholders. We conducted a series of semi-structured interviews with commercial shrimpers, 12 to be specific, whose experience cumulatively came to almost 400 years in the shrimping industry. These were generally captains of vessels. Some also owned processing facilities, and they worked sort of all over the Southeast, but had home bases in either South Carolina or Georgia. And three main themes popped up from the interviews. These were focused around the environment, changes in shrimp, and the need to be adaptable as a commercial fisher. I'm not going to read these quotes, but I'll leave them up here on the screen for just about 30 seconds so that you all can see sort of what we heard from industry. So one of the other questions that we asked, or one of the questions that we asked the shrimpers was, what are your top three concerns for your industry moving forward? And perhaps somewhat naively, we weren't expecting that their main concerns were really socioeconomic as opposed to sort of environmental factors. So what we heard a lot was that competition with imported shrimp was a problem, potential for stagnant or even falling prices for their product, coming up against rising operating costs and aging infrastructure. One thing we also heard that might beg some future work is this increase in interactions with sharks as populations of sharks in the coastal zone have increased over the last 2 or 3 decades. They are attacking trawl gear and leading to a lot more damage to fishing gear than shrimpers had seen historically. So we didn't structure these as sort of a true social science formal survey. Instead, what we really got out of this was sort of contextual information for the researchers on the project about how can we, as we interpret our environmental science data, let's keep in mind what the industry is facing and what the folks really on the water every day are seeing, and how do those two sort of data streams line up. Really quickly, I'm going to cover, or just let you know that we have recently published a paper on the laboratory experiments conducted through this project, where we manipulated density and species composition of shrimp in some mesocosm tanks in the lab. You can find that paper in PLOS One. And this was an opportunity mainly to engage a lot of undergraduates in the research process. In terms of field surveys, this is very much ongoing. Our field collections are complete, led by Liam and another graduate student, Caitlin Glover. That crew with local partners including Graham, sampled every month for the 6-month summer period over 2 years across 10 sites in the 3 reserves. And so at each one of those 10 sites, we collected larval shrimp and other animals using the epibenthic sled that Josh covered. We collected subadults and juvenile shrimps and all the other critters in the creek using trawls and cast nets. And we collected benthic infauna, which really are the prey base for shrimp, using a core sampler. And these are just some nice action shots of the team in the field. And here's some of the key critters that we encountered. We are conducting a number of analyses on these data sets. We're really excited about this sort of regional look at tidal creek communities. Some of those include just looking at the larval fish community across these estuarine systems, looking at potential for top-down control of shrimp by predatory fishes. One of Liam's dissertation chapters is a snapshot of nekton use of tidal creeks and how that changes with environmental conditions. And then we're going to be linking those shrimp life stages and their prey base with the environmental conditions that were occurring at each one of our sampling sites, because we designed this study to utilize the swamp infrastructure in each of the 3 reserves. Really quickly, because we're running out of time, I'll just cover our outreach with industry folks. About 2 months ago, we held a shrimp social in Port Royal Sound in South Carolina, where we invited about 30 shrimping industry end users to come and hear about the research outputs. We shared a delicious shrimp dinner that was catered by one of our local commercial shrimpers, and we had the opportunity to get feedback from the industry on the work that you've heard about today, and also talk about potential ways to collaborate moving forward. So we've heard nothing but positive responses from the industry folks about this event, so we're really excited about continued interactions. The other hot off the press outreach product that we've generated just recently is a story map. So most of y'all are familiar with these probably, but this is an interactive web-based framework for sharing information. We've developed this in collaboration with the Georgia Sea Grant Communications team, and so we're really excited about this as a way to share our research outputs. I'm going to add the link in the chat here shortly. So this has been a really productive group. We've published 4 papers so far. We have another, I'd say, 6 to 8 papers in various stages of the publication process. I say that mainly to point out that the NERRS Science Collaborative webpage associated with this project is serving as a repository for all of these papers. So if you're interested in learning more, you can find all the existing and the future papers associated with the Lowcountry Shrimp Collaborative at that website. So you met 4 of us today. We're a really big team. I have to acknowledge all the rest of the Lowcountry Shrimp Collaborative core team, as well as the numerous undergraduate and volunteer researchers who have helped out on a variety of aspects of this project. We appreciate the funding support from the Science Collaborative, and I'm available for questions if we have any time, and I think the panel is still here as well. So thanks again for joining.